So just to review the homework assignment that you had, the Amoeba Sisters video recap. The first one, pretty self-explanatory. Why does DNA need to replicate before cells divide? To make sure that the daughter cells have the same amount of DNA if we're talking about mitosis. Or if we're talking about meiosis, right, we need DNA to duplicate so that way when the cells go through meiosis and then create 4 haploid cells, that they each have 23 chromosomes like they're supposed to for gametes. So just making sure that the cells, when they divide, have the appropriate amount of DNA. If the DNA did not replicate, then the amount of DNA in the cells would decrease every time that they would divide. Number 2, in the below DNA diagram, label one of each of the bolding items. So you should have tried your best to label the items. Deoxyribose, which is the sugar in DNA, that would be one of these. Total number in the image, 1, 2, 3, 4, 5, 6. Nitrogenous base, so nitrogenous base would be one of these: adenine, thymine, cytosine, or guanine. And there are 1, 2, 3, 4, 5, 6 as well. Hydrogen bonds. Where do you find hydrogen bonds? You find them in between the nitrogenous bases. So hydrogen bonds, there are 1, 2, 3 of them. And phosphate. These are representing, or here, I'll just do this, representing the phosphate. Label 5' to 3' strand and the 3' to 5' strand. You look at the carbons at the top of each strand, this one and this one. It is the carbon that is facing towards the outside, or quote unquote hanging off the DNA. This one would be 5'. This one would be 3'. And that means that this strand, this side would be 3'. This side would be 5'. So the strands should be numbered 5' to 3', 3' to 5'. Number 3, here are all the enzymes, well, most of the enzymes involved in DNA replication. In your own words, describe their function in DNA replication. Helicase, we see helicase is the enzyme that unzips DNA. Unzips DNA by breaking the hydrogen bonds. DNA polymerase, to be more specific, in this video, it only showed you, it only talked about DNA polymerase as a whole. It didn't number them. Specifically, this is talking about the function of DNA polymerase 3. DNA polymerase 3 is the builder. It builds DNA nucleotides for the daughter strands in only the 5' to 3' direction. Primase is an RNA polymerase. It lays down RNA nucleotides to tell DNA polymerase where to start building. Ligase seals gaps between the Okazaki fragments by using phosphodiester bonds. So sealing in the gaps of the backbone between the nucleotides that are, one nucleotide of one Okazaki fragment to the other nucleotide in the other Okazaki fragment. This one, you're narrating the illustrations to explain the sequence of events that occur in DNA replication. So picture number 1, right at the point of origin, you see little enzyme helicase. Helicase unzips DNA at the point of origin. Picture number 2, single-stranded. This talked about it in the Amoeba Sisters video, but there's these single-stranded binding proteins that keep the strands apart, keep them from coming back together. But more importantly, in this step, primase lays down RNA nucleotides. Then next, we have DNA polymerase 3. Lays down DNA nucleotides. So like this picture doesn't do a great job at showing, because it's not, like here it's showing you that primase, RNA polymerase, actually put down a nucleotide primer. Here it's just showing a little flag. Obviously it's a cartoon, but DNA polymerase will be able to build DNA nucleotides off of the RNA nucleotides that primase laid down for it. And in that last picture, so DNA polymerase 3 is building the on that leading strand perfectly fine, right? That would be up here, leading strand. And Okazaki fragments were created on the lagging strand. Why? Because DNA polymerase can only build in the 5' to 3' direction. So the RNA primase had to put several primers down, which creates fragments, Okazaki fragments, instead of having one long continuous strand. So that kind of goes into what number 8 is asking you. Leading strand's the top one. Lagging is this bottom one. What does it mean by lagging? It's moving in the opposite direction. DNA polymerase 3 is building the daughter strand in the opposite direction of the way that helicase is unzipping the DNA. So that's what leads to the creation of the Okazaki fragments. And why does it occur? Again, it occurs because DNA polymerase can only build in the 5' to 3' direction.